All right. So this is how we are going to run animal cell culture modules in next 5 weeks or so. So there are 5 lectures, you can say. So first lecture, which is today, is introduction to the art and science of animal cell and tissue culture. I say this is first art and then science. And I will try to convince you when you are working in the lab, you will see it's more of art. And when you start working with your result, then the science comes. So when you are working in the lab and setting up experiment, that's art of the tissue culture. And once you start getting your data from those experiments, then you come in actual science. So working in the hood is mostly art. And then once you have completed your task and then you are going to acquire results, then science comes in picture. Lecture 2 will be more details on technical aspect because it's a vocational focus course. So mostly we will talk about the techniques, how. And then third and fifth would be mostly like more details on the technique, and then we will go in a specialized applications of the animal cell culture technology. That's how I have planned. Let's see. So in this lecture, what we are going to cover, and I would be rather interactive. So I expect like we will have a discussion, and you feel free to write into the chat boxes whenever you want, and then we will start discussion from here. So today we will cover what is the tissue culture actually, how old is this science, and little bit of the history. Then why do we need to do cell cultures on the first place? What are the basic requirements for tissue culture? And then some of the properties of the cells which are grown in tissue culture, or you can say in Petri plates. So this is an activity. If you see on your screen, you see there are three uh, figures, and try to identify and write what you see in here in these upper two and lower one. And can you identify any of these? Say something about this. Okay. Think about on the cellular aspect of this. Okay, so make some observation. What do you see into the top green panel and the right hand side panel? What do you see? Yes, typhoid, vascular bundles, and parenchyma. Hmm. How do you know this is vascular bundles and parenchyma? Steel of a plant. Good. There's another view which I wanted to put a viewpoint on here, which is kind of you were very correct. The upper two panels were from the plant, and lower one is from the kidney. Okay. The observations are. If you see in the plant, if you see, you can find definite structures in this, right? Definite structure on top too. You see some geometries there, which are cell structures are like you can make out these are separate cells. But in case of down figure, it's very difficult to find out an individual cell or make it like which will be the individual cell, because there is a histopathology slide which has a purple color or you can say blue color nucleus. So you can say, oh, these are the cellular nucleus, but you cannot make out exactly that boundary. One problem. Second is in these figures don't show any scale, right? So it becomes very difficult for you to interpret whether it's a plant cell or plant part or animal cells, because I have intentionally not put a marker scale. So it becomes very difficult for you to. So, so there are, the reason here is just wanted to make some general point. Whenever you are putting your data, you make sure that if you are measuring using microscopic images, put markers in there and then tell what you observe. Um, Whatever you have said in dicot, xylem, and phloem, that's the secondary thing. First thing is what you are observing in that particular slide. So you are observing some structures which have definite shape, and down it is not. So you start observing that rather than making your like final judgment. So there are something which is distinctive feature is cells in the plant tissues are much more organized than easily identifiable as compared to lower this where you see renal kidney. Can you tell why it is? Why it is so? You all know, actually, you all know. It's just I'm not testing you because of the cell walls in the plant. Yes, that cell wall is the beauty of the plant. That's why you can see is easy to maintain into culture as compared to animal cell culture, and that's why animal cells. What is in animal cell plant? How they maintain their shape? Anyone? Membrane. What kind of this membrane is? Cytoskeleton and membrane. Two things are good. If you know what is the structure or composition of these membranes? Lipid, yes, and it's a lipid bilayer. Precisely, it is a phospholipid bilayer because when you ask a lipid bilayer, lipid might be a triglyceride, and that will not be because that will be only hydrophobic. But if you use phospholipid, that becomes a bipolar molecule, right? So phospholipid and protein. So it's phospholipid bilayer. And again, we call it's a self-assembled phospholipid bilayer. What does it mean? It means if you take a phospholipid and make a drop and put into the water where the surrounding is water, this phospholipid will self-assemble itself in the form of a micelle. So it's a self-assembly. You remove the water, the integrity of the phospholipid micelle or the cell will go. So it is very important that within and outside for animal cells, it should be an aqueous environment. Make sense to you? While, and then the tonicity also comes in picture. What is tonicity? The relative concentrations of salts across the membrane, okay, within the cell and outside the cell. That's very important in case of animal cells. While in plant cells, what is turgor pressure? So membrane can take up the water and then it expands, but the cell wall does not allow you to it to rupture. But if you put these animal cells into the water, just water, what will happen? They will bulge, and because there's no cell wall, they will burst. Yes. So these are important considerations when you are doing, when you are culturing them into the lab. Okay. So I'm going to give you a quick revision, which is about the characteristics of animal cells, right? So they, if you isolate these cells, they will be ranging from 6 to 30 micron in size, micrometer. So can you guess which is the smallest cell? Yes, red blood cells are by far the, yep. So RBCs are the 5 micron, and then others are roughly in, like if you take in them in solution, isolate them, they will be 10 to 20 micron in size, and then some cancer cells are larger, 6 to 30 micron in size. And which are the largest cell actually in the body, in our body? Can you, any guess? Neurons, perfect. So this is when we are talking about the size domains. The shape is like for animal cell, it's a spherical or ellipsoidal or irregular. You cannot bound them in a particular shape. You cannot say they don't have a definite shape. Okay. In cell structure, because they don't have a definite shape, because they don't have cell wall, only thin and fragile cell plasma membrane, and which is made up of proteins and phospholipids. It's a negatively charged surface which help in sticking to each other or to the some plastic surface or wherever you want to grow them. They have receptors which gives them a key kind of identification of those particular type of cells. And then some animal cells are encourage dependent. That means they if you take them into the culture, they can survive without sticking to any substrate or any plastic substrate, right? So I will use often these words: substrate, substratum, encourage. So this is a kind of a new vocabulary if you have not heard about that, and we will use these words very often. So I'm trying to introduce and we'll go. And then cytoskeleton, right? So animal cells have cytoskeleton which have actin filaments, intermediate filaments, microtubules to provide cells mechanical strength and control shape and guide. So this all, I think you have, most of this you have studied already in your previous year and VCE. So you know, just I was brushing it up. So what is cell culture?